Hi, I'm Will Chou, CEO of Aruvant Sciences. We are a company dedicated to developing life-changing gene therapies for patients with rare diseases. The lead product in our portfolio is Air U1801, a lentiviral gene therapy for sickle cell disease. Air U1801 is in an ongoing phase 1/2 trial and has already demonstrated potentially curative clinical efficacy with up to 100% reductions in vaso-occlusive events and durable responses for more than 3 years. Air U1801 has a toxicity advantage versus other gene therapies in that our product requires lower doses of chemotherapy than other gene therapies. Our second portfolio product is Air U2801, which is an AAV gene therapy in preclinical studies for hypophosphatation. 2801 has demonstrated durable increases in tissue nonspecific alkaline phosphatase through 18 months and is a potential one-time treatment that would replace a chronic enzyme replacement therapy standard of care. We have an experienced team in gene therapy and rare diseases. I previously led the adult clinical development program and the commercial launch of Kymriah, the first CAR T to be approved. And Palani Palniappan, our Chief Technology Officer, has led tech ops for multiple gene therapy programs previously at Sarepta. As a young company, we prioritize bringing in experienced gene therapy scientists in CMC, quality, regulatory, and clinical. And in a remote work environment where talent has more and more choices about where they want to work, we've set up a culture and infrastructure to attract and retain our best talent. I'm going to spend most of our time today speaking about Air U1801, our potentially curative gene therapy for sickle cell disease. Now, the hallmark of sickle cell disease is painful vaso-occlusive events, otherwise known as VOEs. VOEs lead to hospitalizations, they lead to organ damage in sickle cell patients, and also they lead to decreased survival. Patients with severe sickle cell disease only have a life expectancy in their mid-40s. Now, as you know, there are several gene therapies being investigated for sickle cell disease, and each of them have a different mechanism of action and different therapeutic hemoglobins that may each have different intrinsic levels of effectiveness at ameliorating symptoms. As we were taught in med school, we don't treat labs, we treat patients. And the common denominator for every therapy is how are you affecting the patient's life? So in our discussion with regulators, and from what we've seen from other programs in sickle cell disease, that common denominator, that endpoint that matters the most is reduction in vaso-occlusive events. So what is Air U1801? Air U1801 uses a self-inactivating lentiviral vector that contains a proprietary gamma globin gene for a novel, highly potent variant of fetal hemoglobin, hemoglobin F G16D. The unique potency of Air U1801 allows it to engraft with only reduced intensity conditioning. And we have shown VOE reduction up to 100% and durable engraftment over 3 years. So how do we make Air U1801? Well, first, we produce the lentiviral vector. Second, we collect CD34 positive stem cells from patients. We transduce those stem cells ex vivo, and then we reinfuse the transduced stem cells after a single dose of reduced intensity conditioning. Now, a word about conditioning. Every gene therapy for sickle cell disease requires some conditioning prior to treatment. The purpose of conditioning is to reduce the amount of patients' naive stem cells and give space for the transduced stem cells to engraft in the bone marrow. Air U1801 is the only gene therapy for sickle cell disease that uses reduced intensity conditioning. All other gene therapies require a larger dose of chemotherapy to enable efficacy. And why is it that Air U1801 is able to engraft with only reduced intensity conditioning? Well, there are 3 factors that are unique to Air U1801 that allow us to use RIC. First, Air U1801 creates more hemoglobin F per vector copy. The proprietary G16D point mutation drives a higher hemoglobin F payload per vector copy. That single point mutation makes the gamma globin that we encode more negatively charged and therefore is more likely to bind to positively charged alpha globin. And that creates more hemoglobin F per vector copy. The second point is that the hemoglobin F that we make, hemoglobin F G16D, may have a more potent anti-sickling effect than endogenous hemoglobin F. We've demonstrated that in preclinical models, and from our early data, from what we've seen from patients we've dosed, we have similar signals in humans. And finally, there is a proprietary manufacturing component that we add in culture as part of our manufacturing process that enables more true stem cells in each dose of Air U1801 administered. When you put stem cells into culture, they want to differentiate into progenitor cells, and progenitor cells do not contribute to long-term engraftment. This proprietary additive that is part of the manufacturing process, it doesn't touch the patient, it's part of the manufacturing. This proprietary additive it preserves the stemness of stem cells in culture. Therefore, we end up transducing and transplanting more true stem cells for each dose of Air U1801 that we administer. So what does that all mean? When you add it all together, it's these three features together that allows us to get durable engraftment with only reduced intensity conditioning and allows us to get strong efficacy with lower vector copy numbers. Now, we talked about how we can only use reduced intensity conditioning, but why is that important? Well, we use melphalan 140 milligrams per meter squared as our conditioning. Other gene therapies for sickle cell disease use fully myeloablative doses of busulfan. And what we've shown in this chart is the toxicity differences between these two regimens. And these differences have a major impact for patients, providers, and payers. First, with melphalan 140, you have a shorter duration of neutropenia and thrombocytopenia. Therefore, you reduce that period when the patient is most susceptible to infection or sepsis or bleeding, such as intracranial hemorrhage. With melphalan 140, no seizure prophylaxis is required, whereas seizure prophylaxis is required for myeloablative busulfan. The rate of ovarian failure is markedly lower with MEL 140, and this attribute is quite important to the younger patients who are looking to get gene therapy. And finally, administration is only 1 hour and often is done in the outpatient setting. So we know because MEL 140 is a commonly used chemotherapy regimen in auto transplants for multiple myeloma, and these are frequently done in the outpatient setting. Fully myeloablative conditioning requires daily PK monitoring, and most importantly, it requires a long hospital stay. So 4 to 6 weeks inpatient hospital stay for myeloablative chemotherapies. So as you can see, that makes a huge difference to a patient and potentially to payers and hospitals as well. What you see here is some new data that we haven't shared yet, and this is data from the first 4 treated patients with Air U1801. So in the 2 years prior to treatment with Air U1801, all 4 of these patients experienced a significant number of vaso-occlusive events while on standard of care therapy. Each of the red dots on this graph is a VOE which these patients experienced during the baseline period, which for our study is the 2 years prior to receiving Air U1801. I will note one thing, which is the first 2 patients were treated before Aruvant took over this asset. Prior to treating patient 3, we made and continue to make major investments in improving our manufacturing process. And now in this chart, we've added the follow-on period after treatments. So you can see in the 2 years following treatment, patients 1 and 2 had a 93 and 85% reduction in VOEs respectively. This is a significant improvement in their lives, but still not quite curative. At the current time, patients 3 and 4, the first to be treated with our updated manufacturing, have reported no VOEs at 18 months and 12 months of follow-up as of today. While we need to play this out and see how these patients do over time, we are very pleased with the potential of Air U1801 to offer complete resolution of VOEs for these severely affected patients. And finally, how do physicians perceive the market role of potentially curative gene therapy, which uses only reduced intensity conditioning? Well, the data you see here are from market research with hematologists and transplanters. And on the left, this data tells us that in a future market where only myeloablative gene therapies were available, hematologists and transplanters considered 49% of their severe sickle cell disease patients eligible for gene therapy. However, in a new market that had both a myeloablative and a reduced intensity conditioning gene therapy as options, they would now consider 70% of their severe sickle cell disease patients eligible for gene therapy. So by having a better toxicity profile of the conditioning, more patients are potentially appropriate for gene therapy. And of those eligible patients, the majority who get gene therapy would get the reduced intensity conditioning gene therapy, which is Air U1801, which makes sense. A product with the same potentially curative efficacy with a lower toxicity profile has the potential to be a market leader. Finally, I'd like to share some early data about our pipeline program, Air U2801 for hypophosphatation. So Air U2801 is a one-time gene therapy for HPP that could replace a potential ERT blockbuster standard of care and deliver curative efficacy without the limits of chronic administration. So what is Air U2801? It is an AAV gene therapy that in preclinical models has demonstrated durable and sustainable increases in tissue nonspecific alk phos through 18 months. And it shows the potential for significant reductions in disease burden. So increases in survival, 18 months versus 3 weeks, amelioration of bone defects with the development of mature bone, and normalization of body weight and bone density in preclinical models. So what is hypophosphatation? HPP is a devastating and potentially fatal orphan disorder with no gene therapies available. It's caused by a mutation in the ALPL gene, and it's characterized by low or no expression of tissue nonspecific alkaline phosphatase. Without functional ALP, what happens is limited bone mineralization, which leads to severe musculoskeletal compromise. So the severe forms of HPP result in respiratory failure from chest hypoplasia, seizures, and limb deformity. Left untreated, 50% of the most severe type of HPP patients die within the first year of life. Now, the current standard of care for HPP, Strensiq, has made a major monumental difference in the lives of patients with HPP. They went from having no hope to having indefinite hope. However, Strensiq requires chronic administration, and many patients experience injection site reactions. So there is a high unmet need in the market, despite the positive impact that Strensiq has made. So Strensiq requires up to 6 subcutaneous injections per week for a lifetime. And these injections frequently come with adverse events. So 3/4 of patients get injection site reactions. And we did some focus group with HPP patients, and these are direct quotes from some of those patients. I wouldn't hesitate with something new if it meant less injections. The second quote is, every injection is the most dreaded experience. It's like asking to inject fire into your body. Air U2801 has the opportunity to eliminate these chronic issues with injections. And finally, some of the preclinical data from Air U2801. And what you see on the left is preclinical data from a murine model of hypophosphatation. The y-axis on the left is ALP activity, and the x-axis is months. And at the lower left, you can see a single dot, and that is the murine model of HPP untreated. And as you can see, this is a very severe disease, and untreated, these mice die in less than a month. And so that is why you only see a single dot here. And what we also show is at therapeutic doses of Air U2801, with a single dose of Air U2801, we can show marked increase in ALP activity that is durable over 18 months. And on the right side, in this same study, over those 18 months, the overall survival was 70% in these treated animals, markedly improved from the disease model. Importantly, there was no evidence of ectopic calcifications in any of these preclinical models at these therapeutic doses. Looking ahead, we will be continuing to enroll patients in our phase 1/2 study of Air U1801 through the end of this year and into early next year. We will be announcing more clinical updates from this phase 1/2 trial later this year. We will also continue to generate non-clinical data for Air U2801, and we will be initiating a pivotal study of Air U1801 early in 2023. Thank you for your interest in Aruvant Sciences.